Hello everyone, welcome to the ITLS Academy. Today we are going to perform another staining technique, that is your endospore staining. Okay, so before discussing the endospore staining, we are going to discuss about the some basic of the staining. Now staining are what? We all know that staining is a microscopic technique which we used for the enhance the contrast of the microscopic image. We all know that we use a microscope to see the microorganisms, and these microorganisms are what? They are colorless. Okay, so if these colorless microorganisms are seen under the microscope, then it cannot be easy to visualize it. Okay, and the image of that microscope are also not that much quality. Okay, that's why we use a staining. These staining colored the microorganism or highlighted the particular area. So when we see that image inside the microscope, that image are contrast. Okay, so this is called as staining. Now the staining that we are going to perform today is your endospore staining, which is defined as a we all know that the bacterial endospores are what? They are metabolically inactive. Okay, the endospore which are present into the bacterial cell, they are what? They are uh, metabolically inactive, and they are very highly resistant to their structure, which is produced by some bacteria as a defensive strategy against a unfavorable environmental condition. Okay, so what they do, they use a endospore as a defense mechanism. Okay, when they go a unfavorable condition, then if the endospores are present and that bacteria, they will use that endospore as a defense mechanism, that is called a endospore. So here the primary stain is what? Primary stain is your malachite green. These malachite green are what? They are water soluble and they have very low affinity for a cellular material. So a vegetative cell may decolorize with water, and again we use a safranin. This safranin are act as a secondary stain. Primary stain is your malachite green, and the safranin are act as a secondary stain, and they will and also known as a counter stain any cell which have a decolorized. Okay, so the cell which are decolorized by using the distilled water, then it will colorize with the help of that safranin. That is called the that is all about the endospore staining. Now what we have to do, we take a slide, and with the help of this inoculation loop, which are already sterilized by using the flame which come out from the burner, we take the smear and put it into the slide. Then after that we add this malachite green into it. These malachite green are forced to enter into the spore by steaming the bacterial endospore. Now after that we put that slide inside the hot water bath at 100 degrees Celsius for 5 minutes. Then after that we rinse that slide with the help of the distilled water. Then we add a safranin. Then after 30 seconds we again rinse that safranin with the help of the distilled water, and then we examine it under the microscope. The vegetative cell appear pink in color and the endospore appear green in color. Okay, that I already told you. This malachite green are what they are water soluble, and when we use our distilled water for a rinsing purpose, then if the endospores are not present, then it will take out from the distilled water. Okay, now let's start the experiment. First of all, take out the slide. Okay, now also we have what we have a culture which we perform by a pour plate method technique. Now how we do these pour plate method technique, and how we first of all we have to prepare a media, then we autoclave that media, and then we do a serial dilution. After serial dilution, we sterilize the laminar airflow, and then we perform a pour plate method. How we do all these experiments, videos related to these experiments are already uploaded in our YouTube channel. You can visit our YouTube channel and see that videos. Okay, so then after that you can better understand what we do. Now with the help of these inoculating loop that I already sterilized in front of you at the starting of the video, so that it will be cool down, because if we take during a heat purpose, then it will damage the bacterial cell. Okay, now put it into the middle of the slide. Now first of all we are going to add a primary stain, that is your malachite green. Then we put this slide into the water bath at 100 degrees Celsius for a 5 minutes, so that this malachite green can enter into the spore of the endospore of the bacterial cell. Now after 5 minutes, what we have to do, we have to wash it with the help of that distilled water. Okay, then after that what we have to do, we have to air dry it, and then we add a secondary stain, that is your gram safranin, or sorry, that is your safranin. Okay, now we wash the stain with the help of the distilled water. Now I am going to add a safranin into it, and after addition of the safranin we have to only wait for a 30 seconds. Then after 30 seconds we wash it with the help of that distilled water. Then we examine it under the microscope. Now the image that we take with the help of that microscope will be attached at the end of your video, so that you can better understand that how, which type of image that we get when we see our slide inside the microscope. If the vegetative cells are present, no, sorry, the vegetative cells appear a pink in color, and the endospore cell appear a green in color. Now wash it with the help of the distilled water. Then after that we have to air dry it, so that this water will remove. Okay, you can also use a tissue paper for the absorbance of that water droplets. So this is all about the endospore staining. I hope you will understand, but if you have any query related to this experiment, you are free to ask me in a comment box, or for a further detail you can follow the ITLS Academy on different platforms, that is your Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn, and subscribe our channel ITLS Academy on YouTube, and get our app from a Google Play. You can also visit our website, that is your www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.